Yeah, thanks for everyone who came. I see a lot of new faces here. That's good, and I hope you guys can now, be part of Csaaw and receive the emails and coming and you can order food, these cool things. So we have JK today, and he is from the math department here. That's like math, and he's gonna be talking about the, as you can see, the problems with biological clocks.
Thank you for giving me a chance to talk here. Actually, I'm a PhD student in math department, and my research has focused on biological clock in our body. Today, I'd like to discuss about two central problems in biological rhythm. First is how can we understand the complex network function? Second, how can we identify the structure of this biochemical network? So first, let me introduce what is our biological rhythms. So there are various types of biological rhythms exist in your body. First, your heart is keep beating now, right? So it generates an oscillation about every second. And these heart beatings are coming from heart pacemaker. This heart pacemaker generates the action potential every second. This action potential causes the heart to keep beating. And second, the cells in your body also keep dividing with about every hour. So in that way, you can recover all the died cells. And furthermore, another thing is circadian clock. So circadian clock locating the hypothalamus in your brain. This circadian clock generates the 24-hour periodic clock gene expression. So here is a movie of the circadian clock. Although it looks very fast, it is a 7 days recording. Oh, sorry. Okay, this is 1 day, second day, third day, fourth day.
So when it's bright and when it's active?
So this is this light is kind of represent the concentration of the clock gene. So kind of they detect some luciferase to the certain clock gene, and if they become high expression, the light become brighter. So it looks hard, but it is a circadian clock in your brain. The right side, I think I brought laser point. Okay, thank you. So this is a record of the circadian clock. You can see this beautiful 24-hour gene expression. So this oscillation basically tells the time of the day to our body. So this circadian clock controls lots of things in our body. For instance, lots of hormone secretions are released according to this rhythm. For instance, this is a rhythm of melatonin. So melatonin secretion is going up during the night and going down during the day, and going up during the night and going down during the day. So as well as this melatonin, some serotonin, and various kind of hormone secretions are oscillating according to the circadian rhythms. And your body temperature also shows a 24-hour periodic rhythm. And moreover, your sleep-wake behavior is also showing the 24-hour periodic pattern, right?
Jay, so I have a question about jet lag.
Yeah.
So when jet lag happens, that the period is not 1 day, it's like maybe half a day or something like that. So it's shifted.
It's interesting. Good question. So this is if the circadian clock can communicate with the light-dark cycle. So kind of this light can phase advance or delay. So kind of it can adjust the circadian clock phase. So basically, to recover the 1 day time difference, you need to recover the 1 hour difference, you need a 1 day. So if you go to China, then 12-hour time difference, you need about 12 days.
Wow.
And if you go to California, 4-hour difference, right, you need 4 days to recover your circadian clock. So
Also, don't know if that's what you're going to talk later, but are most of those things related to reactions, and this is the time of some chemical reaction?
Yes. It is a result of biochemical network reaction. Yeah. That is our major talk. Yeah. And another thing is your sleep-wake pattern is also controlled by the circadian clock. So this is a record of the mice activity. So how they can measure this activity is they bring a mice and they put the mice into the one big cage. And in that case, there is a sensor which can measure their activity. So what they do is they turn on the light for 12 hours, which represent the day. And then they turn off the light for 12 hours, they repeat it. Day 9. Because mice is nocturnal animal, so during the day they sleep. During the night, they get up and they eat food and they doing some activity. So wake up, sleep, wake up, sleep, wake up, sleep. But interesting thing happening is when they turn off the light for all days. So that means there is no environmental information about the time. As you can see, there is no more light-dark cycle, but their behavior is really very tightly regulated, right? So sleep, wake up, sleep, wake up, sleep, wake up, sleep, wake up, even under dark cycle. And second interesting point is they get up earlier every day, right? Yeah, they get up about 0.3 hours earlier every day. Actually, the reason is the circadian clock's period of mice is 23.7 hours.
Okay.
Yeah, that's why they get up 0.3 hours earlier every day. What about human? Actually, human circadian clock is not 24 hours. Period is 24.2 hours. So if we do the exactly same experiment for us, actually they did. Harvard Medical School, some 20 years ago, they put lots of undergraduate students to this kind of, not the cage, kind of very room, and they measured this activity. Every day they get up 0.2 hours later. Yeah. So indeed, the circadian clock controls a lot of important physiological aspects in our body. If heart stop, we die. If cell does not generate cell division, will we die? If circadian clock does not work, you get up 2 a.m., sleep. It's really disaster. So there are kind of more various kinds of circadian clock, biological clock exist in your body. We can categorize them according to the source of rhythm and tunability of period. So what is a source of rhythm? So there are two types of biological clock which can generate rhythm by themselves, or certain type of clock can generate the rhythm only when some output stimulus are given. So the most well-known example of this categories are neurons in your brain. So here are examples. So this is an oscillation of actual potential in your neurons. So this is how we apply the external current in your neuron. So they split out the neuron. So without any external current, they are under rest state. They doesn't do anything. But when they apply some external current, if the external current is too small, then this neuron doesn't show any oscillation. But the strength of the stimulus are strong enough, then it begins generating this rhythm. So that's why these neurons are called exogenous rhythm. And here are another type neuron which also show when the external stimulus is too weak, then there is no oscillation, but the stimulus are strong enough, it can generate this type of rhythm. But you can see some difference between this type 1 and type 2 neuron. Can you see something? So, as strength of the stimulus are changed, type 1 neurons frequencies changes a lot, right? It is slow, but here it's very fast. However, type 2 neuron, their frequency is almost same. So that's why we call type 1 neurons period is tunable, but type 2 neurons period is robust. So For last couple decades, mathematicians tried to understand why neurons can show this different type of behavior. It turns out this type of neuron shows a snake bifurcation, and this type 2 neuron shows some hop bifurcation as the strengths of this stimulus are changed. So if you, I don't have enough time to explain this bifurcation thing, but if you search the type 1 or type 2 neuron in Google, you will see the snake or hop bifurcation things. Okay, now let's discuss about this left side. So this clock can generate rhythm by themselves. For instance, your heart. We don't do anything, but they can keep beating, right? And this cell cycle, also cell keep dividing by themselves. And circadian rhythm also, if you extract the circadian clock from your brain, and if we culture them, they can maintain rhythm for some couple months by themselves, if the nutrients are enough. But these clocks are tunable, but these clocks has a very robust rhythm. What I mean is, if you imagine your heart beating, the frequency of your heart is constant? No, right? When you sleep, it's slowing down, but when you do exercise, it becomes faster, right? So depending on your body condition, we have to tune this period, right? This is very essential property of this clock. However, circadian clock, they has to maintain 24-hour period. So if we culture the circadian clock for 6 weeks or 8 weeks, the period 24 hours are really tightly maintained without some environmental cue. So there are 2 types. One is tunable clock, another is robust period clock. So let me discuss about this tunable clock, how this class can generate the rhythm such a wide range of frequency. So this clock has this kind of very complex biochemical network, which consists of proteins and mRNA and genes. So they interact with each other and they can generate certain rhythms. So it looks very complex. Not only these 2, there are some 3 or 4 more classes in this category. All of them has one common structure. First, it has some negative feedback loop, which means activator promotes the production of certain repressor, and this repress activator so that it can stop their production. Actually, this is very trivial because to generate the rhythm, we need somehow negative feedback loop, right? For instance, let's say R is going up. To generate the rhythm, we need some pushing down this one, right? How? Through the negative feedback loop. And then if it is going down and it is too small, then the negative feedback loop will be released and going up. So going down, going up. So kind of this is trivial. All of biochemical network need at least one negative feedback loop to generate rhythm. But a couple years ago, the James Farrell lab in Stanford University found that this class of clock has another common characteristic. It has all of this structure has additional positive feedback loop here. Activator activate itself. So here, fast means this positive feedback loop has some faster time scale than this negative feedback loop. This means the degradation rate of activator is much faster than the repressor. So what I mean is, the degradation rate is faster means it can reach the steady state quickly. So that under control of certain feedback loop, if the component has a faster degradation rate, it can respond more in the fast time scale. So all of these class have this structure. So this James Farrell lab group wonder, theoretically, this single negative feedback loop is enough to generate rhythm. But why nature has this one more positive feedback loop? So to answer this question, they compare these 3 structures. So just a single negative feedback loop, a negative with positive feedback loop, and double negative feedback loop structure. How they compare is, they randomly select a parameter for these 3 structures. because this can be changed to a certain ODE model, and they randomly choose the parameter, and they measure the chance of rhythm occurrence of these three structures. As a result, this green bar shows the highest chance of the rhythm occurrence. So this green bar is with positive and negative feedback loop structure. So this structure has the highest chance of rhythm occurrence for random parameter selection. That means this structure can generate rhythm over the widest range of the condition, right? Because biology is not some mechanics. We cannot maintain the constant condition, right? Your body temperature changed a lot. As temperature changed, biochemical reaction changed a lot, right? So it is not some constant condition. So over a widest range of condition, this negative positive feedback loop can generate the rhythm with the highest chance. Furthermore, what they found is the frequency also tuned a lot for this structure. So they found that this structure is optimized to generate the robust rhythm with some wide range of frequency. So this is really good design for some heart or cell cycle which need to generate the rhythm with a wide range of frequency. So this study is published in Science a couple years ago. This got lots of some spotlight because this study confirmed some hypothesis or dogma of system biology. So lots of system biology papers claim that the structure of biochemical networks are optimized as a result of billions of evolution to execute their function, right? So our body or some fungus or some fruit fly, they are some result of these billions of years of evolution. So this biochemical network should be somehow optimized by the nature selection, right? So this study explain this dogma of system biology. Then what about this one? So this circadian clock locating various organisms. Actually, almost every organism has circadian clock. Of course, human, mice, cat, lion, tiger, and fruit fly, fungus, some unicellular organism also has this circadian clock. So the common structure of all these organisms' circadian clock structure is double negative feedback loop structure. Additional negative feedback loop with additional negative feedback loop. So similar, there are some couple previous computational study which is studying what is the role of this additional negative feedback loop as the James Federal lab did. Actually, the results are actually bad. Yeah.
Say quickly, how do someone go to measure that you have a negative with a negative feedback?
So if you look at this biochemical network, you can find some negative feedback loop here, and you can find some additional positive feedback loop. So if you look at the circadian clock diagram, you can see some negative feedback loop with additional negative feedback loop. So we simplify this complex network to this simple model. But the previous study found that this double negative feedback loop structure is actually bad. You really easily lose rhythms. That means single negative feedback loop is much better than this double negative feedback loop structure. This is really not a good news because experimental study claim that circadian clock has this double negative feedback loop structure, but computational study claim that this double negative structure is bad. That raised a couple questions. First, this dogma of system biology does not work in this case. The result of billions of evolution, this double negative structure is bad. Or second question, maybe these experimental studies are wrong. Somehow there is some unknown or hidden feedback loops. Or third then what is the design? Good design for this clock with the robust period. So these three are the questions I'd like to discuss for next chapter. And this study based on this mechanism for robust circadian timekeeping paper, which will be published soon. So what I noticed is, although in biology or chemistry, they usually write the negative feedback loop in this way, right? But this simple negative feedback loop can be a mechanism for this repression varies a lot in biology. There are various mechanisms which can generate this negative feedback loop. So one well-known thing is called some cooperative binding or Hill type equations. So how this works is some mRNA is transcribed and it translates to the protein and it enters the nucleus and it binds to the gene promoter in multiple sites. And when this repressor bound to all of the sites, then this transcription is stopped. So in this way, we can generate some single negative feedback loop. So this number of binding site we call some Hill type coefficient. And this is one way to generate a negative feedback loop. Another way to generate negative feedback loop is protein sequestration. So similar, mRNA is transcribed, it translates to the protein, and it enters the nucleus. Now it bound to the activator, and this bound activator becomes inactive. In that way, we can generate some negative feedback loop. So there are actually 2 or 3 more kinds of mechanisms to generate negative feedback loop in biology. So, but kind of these 2 are kind of well-known example. So what I noticed is all of the previous studies, there are some 3 or 4 previous studies which studying the double negative feedback loop structure. They all focus on this mechanism. The reason is, I don't know if you're familiar with this Goodwin oscillator. So if you look at any mathematical biology textbook, and if there is a chapter called some biological rhythms or clock, this is a first example. Kind of this is developed some at 1960 years ago, this model. Kind of this is used a lot. So all of the previous study actually use somehow different model, but all of them based on this Goodwin oscillator or this Hill type equation. But what I noticed is in the natural circadian clock, the repression mechanism does not work in this way. They use this protein sequestration. So in natural circadian clock, they use this protein sequestration to generate the negative feedback loop. So I just simply changed this equation to here to represent this protein sequestration. So the other equations are exactly the same, but this is pretty complex, right? That's why previous study prefer this type of things. Kind of, if you imagine you do some analysis for these 2 systems, this is kind of some homework problem, but this is really a hardcore problem. So, but I changed it to according to some biological literature. And then I compare structure. This is a core negative feedback loop, and I add additional negative feedback loop or additional positive feedback loop. So, and all of these 3 structures, double negative feedback loop and negative feedback loop and positive negative feedback loops, show some similar behavior for their normal parameter. And then I perturb the parameter, shows which one is the most robust. Actually, surprisingly, this is a range of parameter where rhythm occurs. Double negative feedback loop structure, the blue, has the widest range. But positive negative feedback loop has the narrowest range. Exactly opposite of the previous study. So previous study, which based on this Hill type equation, claimed that positive negative feedback loop is robust. But I just switched the mechanism to the protein sequestration. The results are exactly opposite. So I wonder this is only work for this simple model. So I test for other model. So this is a mammalian circadian clock model, very detailed model with about 200 equations. And this is a Drosophila fruit fly circadian clock model. The common characteristic of this model is none of these models use Hill type equation. So as a result, actually all of this model shows that double negative feedback loop structure has the widest range of parameters where rhythm occurs. Indeed, double negative feedback loop structure is most robust structure. So then what about this one? The positive negative feedback loop structure become robust when this additional positive feedback loop is faster, right? So what about this case? I changed the time scale of this additional negative feedback loop, And as you can see, as it becomes slower and slower, it has the widest range of parameter where rhythm occurs. That means we need some slow additional negative feedback loop. That means activator should be degraded slower. Interestingly, here is some experimental data. So this is a half-life of repressor and activator in mice and Drosophila circadian clock. The top is a half-life of the repressor, and bottom is a half-life of the activator. So this half-life of activator is significantly longer than repressor, right? Indeed, in nature circadian clock, this additional negative feedback loop is really slow. Yeah. Any question about this? Okay. So here is some summary. So from the cell cycle curve, previous study found this as a good design. And from the circadian clock, I found this is some good design. Then next question is, okay, based on Hill-type equation, this can generate the robust rhythm. And based on protein sequestration, this can generate the robust rhythm. Then question is, why there are two different designs exist in biology? Why they do not optimize to the one robust structure? Actually, the difference is frequency. So as you remember, the positive negative feedback loop structure, if you change the parameter, period changed a lot because this is good for tunable period, right? However, this structure, as you can see, for the over wide range of parameter change, period is nearly constant. So this is really good design for the tunable clock, but this is good design for the robust periodic clock. So it seems there are two different designs exist in biology, which is good for robust clock with the tunable period and another with the fixed period. And I searched some experimental literature and I found this. This is a circadian period of the mice. So this is a multi-mice, but what they do is they partially remove this additional negative feedback loop because there are two genes which contribute this additional negative feedback loop, but if we mutate both genes, the mice are died. So we cannot mutate both of genes, but we can only mutate one of the genes, so we can reduce this additional negative feedback loop. As a result, the frequency distributions are varied, right? So indeed, this structure seems important to maintain the robust period. The next question is, okay, this is okay, good design according to simulation. Then what is the underlying mechanism? How they do that? So this is the next question. To answer this question, we need to understand what is a mechanism to generate what is a necessary and sufficient condition to generate the rhythm here. Some true mathematical analysis. For this type of model, Hill-type equation, to generate rhythm or to generate oscillation, we need some steady state become unstable, or eigenvalue should be some positive thing. So for this one, necessary and sufficient condition to generate rhythm is this number of binding sites should be greater than 8. We need a multiple binding site. However, for this type of model, which is based on protein sequestration, we need a one-to-one stoichiometry. Here, what is stoichiometry is, there is a repressor P and activator. The ratio between amount of P and activator should be similar. So this is a requirement to generate the rhythm for this type of model. So, and this is some experimental result. So what they did is, they cultured the mice skin fibroblast. Even skin has a circadian clock. What they found is activator amount is much more than repressor. So activator and repressor is not one-to-one. So what they did is they overexpressed this repressor so that match the amount of activator and repressor. And here is a result. When they overexpress the repressor, the amplitude is doubled. Indeed, this balanced stoichiometry is important to generate the rhythm. And then what is the relationship between them, this balanced stoichiometry and feedback loop structure? Here is the result. So what I did is I perturbed the parameter and I measure how much the stoichiometries are changed at these three types of structure. As you can see, for the same parameter perturbation, double negative feedback loop structure shows the smallest change of stoichiometry, which means double negative feedback loop structure is best at maintaining the balanced stoichiometry. So for the parameter perturbation, this double negative feedback loop structure can maintain the one-to-one stoichiometry. So simple explanation is here. Let's say at the normal state, activator and repressor amounts are exactly the same, so 1-to-1. But for some reason, let's say activators are decreased. So that means there is two negative feedback loops. The second negative feedback loop will become weaker because activator amount is decreased, right? However, repressor amount is now much more, so it will become stronger. So what is the result then? This become weaker, that means activator will be increased. However, the stronger negative feedback loop will decrease the amount of the repressor. So it restores the one-to-one stoichiometry again through this double negative feedback loop structure. So here are summary. So based on cooperative binding, this positive negative feedback loop structure is good design, and it is good for tunable period. And based on this protein sequestration, this double negative feedback loop structure is good, and it is good for robust period. So this gives us one lesson. In the biology, both mechanism of the feedback loop and structures are important to determine their function. Because most of study only focus on structure. But as you can see, depending on how this repression works, the result could be opposite. So yeah, this is end of the chapter 2. Now let's discuss about some new topic, how we can identify a biochemical network. So although I show very simplified negative feedback loop structure, real biochemical network structure looks like this, very complex. So you asked if this are circadian clock biochemical network, you can see there are some couple core negative feedback loop here, right? And there is additional positive feedback loop here. That's what we call negative feedback loop here, so negative and double negative feedback loop structure. So how biologists identify all of these interaction between protein and protein or gene and protein? Are they using kind of this type of tools? But this type of tools are based on try and error. What they do is they select two protein, looks good, and they test it. If it works, that means there is one arrow here, but it does not work, then you have to do it again. But it is very complex procedure. Basically, this one arrow is one paper because it requires some couple years of work of one lab. So that means actually this biochemical network is still ongoing. This is not complete. Every year they publish some new component. So identifying biochemical networks are so difficult. It requires tremendous amount of work. However, output of this biochemical network is relatively easy to measure. So what I mean is, what is the output of the circadian clock component? So all of these components generating the rhythm as a result of biochemical reaction. Through some microarray analysis, we can measure all of this thing, clocking expression simultaneously. So this each row represents each gene. So yellow means going up, blue is going down, going up and going down through the day. So they can measure thousands of genes rhythm simultaneously, but they don't know how these rhythms are generated. And so this tells us identifying biochemical network is very difficult, but measuring the outputs are kind of easy with current technique. So this raised the question. Then we know that this is very difficult, this is easy. Then can we use this output information to reveal network structure? If we can do it, it's really good, right? Measure time courses of all of the genes in our body, and we can tell that these two are interact each other or not, depending on their output. It will be cool, right? So this is a question I do like to discuss for rest time. So we can simplify this question as like here. Let's say we have two components. We can measure the time course of these two components. From these two time course data, can we say something about their relationship? Are they interact each other or not? This is the question. From their time course, can we identify the biochemical interaction between two species? So we can formulate this as an existence problem of mathematics. So here we use very simple but very general model. So the rate of change of our species consists of production and degradation. Production depends on another species S, and degradation depends on their own species R. So this is kind of common model used in mathematical modeling. So what we do is now we have two time courses, and these two time courses are kind of solution of this model, right? So for given two solution, we can test any f and g exists so that this model can have them as a solution. So again, let's assume that two time courses are given. Then can we find f and g such that this model can have a solution as these two time courses? That means there exists f and g. That means there is some relationship between s and r. However, if we cannot find any function f and g which can have a solution r and s, that means there is no interaction between r and s. I know it is a little bit confusing. Is it okay for you?
When you say if there is a function f and g, can you analytically show that it's possible to find those functions? Or
kind of finding the function is a different problem, actually.
Okay.
Here we just determine there exists or not. So to find the function, we need to do some feeding or something like that, right? But most of the time, what we do is we just do the feeding, right? But before that, what we have to know is there really exists a function or not, right? Kind of, yeah. So the existence problem will tell us really the existence of this biochemical interaction. So here I assume that time courses are exact, without no noise, and smooth, and they have some same period, for instance, 24-hour period. So to answer this question, these simple but fundamental functions are essential. It looks complex, but very simple. It is kind of middle school function. So, can I write it? Is it true?
I guess so.
It looks so fancy, so.
Doesn't look like a wall.
Okay. So if there is some function, let's say cosine function, this is a one-trait of the cosine function, then I define a head map. Head map is sending the one time point to another time point where their values are same. So -1 will become 1, or 1 will become -1, and 0 is maximum, so 0 will become 0. So head map of cosine t is -t. Just change the sign of time. But what about the sine t? Sine t looks like this. So one time point here will go to here, and this time point will move to here. Does it make sense? So sine t has some symmetricity here, and symmetricity.
Just like even and odd functions.
Yeah, something like that. So this is a head map of any time course. Actually, not only this simple function, whatever I had some weird shape of time course, we can always define this map, right? Because we can always find the values are same, right? So this is so now I will call this is a head map of the given time courses. So then there are two time courses are given, R of t and S of t, that we can easily find their head map. For instance, the head map of this blue time course is this blue curve. For instance, 10 and 20 have around same value, right? So 10 will go to the 20. For the red curve, 5 and 15 have the same value, right? So that red map sending the 5 to the 15. So changing this 2 time course to the head map is very easy, right? Okay. So if you do this on MATLAB, they will do it in 0.1 second, something like that. And then we composite them. From now, we call pi as composite of these 2 functions. Okay. So there are 2 characteristics of this pi map. First, the domain and range are same because it is from the one period to another period. And second, it is one-to-one map. So that means we can iterate in this map, right? Pi n. So here is a most important theorem. This is a necessary condition for existence. Basically, what this tells us is we iterate this pi map, and if there is any fixed point, then the model does not exist or biochemical network does not exist. So fixed point implies no interaction between 2 species. The reason is here. Let's assume that pi has fixed point t0. So that pi of t0 or S hat of R hat of t0 equal to t0. What this means is, let's say R hat t0 is t1, then this will become t1, so S hat t1 equal to t0. Everyone is okay up to here? Okay. Now let's look at this figure. So t0 will go to t1 by R hat, right? So that means t0, t1 have the same value for this time course. That is the definition of this R hat map, right? And then this t1 will come back to the t0 by Sn map, where S of t has the same value. Good? So from this figure, what we can get is both R of t and S of t has the same value at these 2 time points, right? So R of t and S of t has the same value. That means whatever g, whatever f, if we plug in this value to the z or f, they have still the same value, right? So now let's assume that there is this model. So what this means is, if you plug in this value to here, the derivative of t0 and t1 should be same, right? Because f and g are same at these 2 time points. Does it make sense? Now let's look at the left side. The derivative of r at t0 and t1 can be same. The sign of the derivative of r here is positive and here is negative. The sign should be always opposite, right? So that means this derivative cannot be same. That means this assumption is wrong. That's why the fixed point implies the model does not exist. Okay? But actually, here is one pole. Actually, although the sign over here are different, they can be same. Very extreme case. Although the sign is different, when they can be same? That's 0, right? Exactly. So, kind of to exclude this extreme case, we need this second condition. But in practical point of view, actually, we don't need it. Indeed, really, in most cases, the fixed point implies the model does not exist or interaction does not exist. Now let's apply this theorem to the toy model. So here is a that biochemical reaction network consists of the single negative feedback loop. So we convert it to the ODE system. Maybe you're now familiar with this one. This is so-called again Goodwin oscillator, the famous one. And we simulate their time process. But my question is, let's assume that we have no idea about original structure. That means any pair can have somehow potential interaction. The only information we have is their time course data output. By using this time course data, can we recover this original structure? That is our question, right? From the output, can we say something about their interaction? So here is the way. Now we have time course data. First step is we change it to the head map. So every time course has its own head map, right? So there are 5 head map. Now next step is we have to generate pi map. Pi map is composition of this head map, right? So there are 5 head maps, so there are 5 choose 2 pairs, so 10 pairs. So there are 10 kinds of head map, right? And then we iterating them and see whether there is a fixed point or not. That's all. This is a very simple algorithm. So again, finding the pi map, 10 pi map, and checking whether there is a fixed point or not. And here is the result. After second iteration map, the pi map from the 2 pair have a fixed point like this. There are some fixed point, right? This is our fixed point. Fixed point means 20 becomes exactly 20. Okay? So if you look at which pair generate this fixed point is MMP3 and P1, P4. If you look at the diagram, MMP3 and P1, P4, really they cannot have any biochemical reaction network, so we can exclude those 2 cases. And then after third iteration map, you can see a lot of some fixed point from all the pi map which generate from these 3 pairs. So MMP2, P1, P3, P2, P4. If you look at these 3 pairs, this inside. So again, we can exclude these 3 interactions. So by using just time course data, we can recover original structure completely. So this is a single negative feedback loop. And what about 2 different systems? Now we have one negative feedback loop. This is another negative feedback loop study system. So they are kind of 2 independent oscillators. So this left side is one network structure, and right side is another network structure. After checking the fixed point of pi map, we can remove some of this unrelated component. After checking second iteration map, we can remove more. After third iteration map, we can keep removing this unrelated component. Indeed, this theorem seems working well, at least for this toy model. From the time course data, can we say something about network structure? So as you can see, all of these theorem is very important to checking the fixed point of this iteration map. But there is a people, because what I assume is the data is exact. But in real biology or chemistry, data is never exact, right? There is always some noise. So to apply this theory to the real data, actually I have to check some stability of this algorithm, or I have to incorporate some statistical method which can filter all the noise things. So this is what I'm doing nowadays. And another I have interest in nowadays is actually this does not limit it to the biochemical network. There are various networks exist in the world, some social network or some population food web network, right? So I wonder from some species oscillation, can we say something about network structure? Might be a really interesting question, I think, yeah. So all of this work is with my advisor, Daniel Forger. He is a professor in math department. And this work is supported by Human Frontier Science Program and Pfizer and Air Force grant. Thank you. Yeah, 45 minutes.
Okay. First of all, very interesting work actually, and this is a comment. Probably you're aware of this, but looking in a general level, I think your work is very similar to theory of control system. Yeah. So where they also study.
Okay. So you next. So they call it as reverse engineering some technique to at the engineering and the control by using control theory recover the structure information.
Yeah. So actually many aspects they study stability, robustness of system through feedback, positive and negative, and also through different mechanisms. And also the last part of your presentation is called system identification in controls theory, actually, where they just measure the output and have the system is a black box. They trace back see what the system is.
So for the first thing, for the first chapter, control theorists actually try to remove oscillation.
Oh, okay.
Right? Because they don't like oscillation. They think the oscillation is a bad thing to control something. But in my question is, we need oscillation. Exactly, actually opposite question. Sometimes we adopt their method because they try to remove oscillation. That means if we have that, that we can generate those solutions. So this is some common things we can have. And for the second question is, in most time control theory, what they do is kind of my study, I don't have any restriction for f and g, right? F and g can be any function because we have no idea about what is that interaction type. But most of control theory already fixed what is this equation. They decide this is some Hill type equation, and this is some linear degradation rate, and there is some parameter, right? And then they try to identify this parameter. This is the way they usually approach this problem.
Right.
But my question is the existence of this equation itself instead of just parameter. Yeah, that is, I think, the difference between their approach and this study. Yeah.
I'm totally impressed by your work.
Oh, thank
It's
you.
really fascinating, really fascinating. Thanks a lot. I'm glad I came here. I do think I understood most of what you said in the last part of the presentation, but I'm not entirely sure. Is there a relatively intuitive way how you can explain me that the non-existence of the two functions of interactions between the two components is sufficient even in such a complex network that you're showing us? I think there's, for me, intuitively, and as I said, I didn't get to the gist of it, I would assume that, so you measure the concentrations of the time series of two components, how do you know that there is not a function that crisscrosses through the network and does something weird with it?
It's really excellent question. Yeah.
Okay.
So kind of this is related somehow like this. So kind of one people of this is we assume that only one-to-one interaction, right? But some may kind of this kind of interaction also exists, right? To describe this multiple interaction, we need some different type of model. For instance, currently we assume that here is only S, but in real life we need S1, S2, some S3, blah blah blah, minus G over R. So we need somehow different type of model to consider this complex thing. Actually, this is what I'm doing nowadays. But kind of similar fixed point theory actually can be applied for this generated model, but it becomes a little bit more complex. So I'm a little bit struggling with that one. And another question is, what if there is some unknown hidden step, right? And is it your question?
Yeah, so I guess when you start excluding relationships from the time series, you still consider only two components at once.
Yeah, just two, yeah. I decide if there are some complex network, I just looking at every pair and see there exists the relationship or not.
So experimentally it would assume, I don't know.
Yeah, it's really very first step paper.
Okay.
Because I don't know how long will it take to really apply to real data, but I hope people,
There is any kind of, when you are applying your model that's iterative, and do you think there is any physical meaning of the amount of iterations eventually? You made a first iteration and like 80% of those connections are gone.
Actually, that is very good question because anyway, we cannot check all the fixed point for all the iteration map, right? So I'm trying to find some mathematical theory which describe about some iteration maps fixed point, but unfortunately most mathematical theory based on topology assume the continuity of the map. But as you can see, the map here is not continuous, it is discontinuous. So I cannot apply all of that previous study. So at this point, I cannot say there is any theory which can tell us where do we have to stop. But when I do some simulation with this toy model, usually 5 or 6 iteration map usually we covered all of the models. So moreover, more than 10, due to there is some numerical error, I think we can get some exact simulation.
So you mentioned that the finding of mouse skin cells, so that you didn't have one-to-one stoichiometry,
Yes.
and when you increased it, it was just amplitude change, not frequency.
Frequencies are about 1 hours are changed.
Oh, it's really hard change also.
Yeah. About 1 hour change. And another thing is, because a fibroblast, rhythms are kind of dampened. Because the master clock can generate the rhythm for forever, but this kind of dampened. But when we increase the stoichiometry, this dampening is significantly increased. Kind of after usually 1 week they dampen, but with this one-to-one stoichiometry, it generates rhythm some couple weeks. Yeah, that is I think the main difference.
Yeah, have there been any studies of, say, the supervised nucleus and the stoichiometry?
Good question. Because stoichiometry is very tiny, so measuring the protein amount is very difficult. As far as I know, no one can measure the protein amount in the supraplasmatic nucleus. So, as far as I know, there are only 2 literature which measure the stoichiometry. One is for the fibroblast and one is for liver. In the liver, the stoichiometry is 1-to-1. Moreover, liver, rhythms of liver does not dampen. It is sustained forever. I think that might be the role of some live one-to-one stoichiometry. Yeah, but I think if we can measure some supraplasmatic nucleus, actually that would be that's the experiment. Yeah. Thank you.